She needs to record.
Oh. Okay. That we introduce our esteemed speaker for today's event. Please join us in welcoming Dr. Peter Koulen, a distinguished figure in the field of ophthalmology research. Dr. Koulen serves as the professor and vice chair for research in the Department of Ophthalmology, holding the prestigious Felix M. Carmen Zabate Missouri Endowed Chair in Vision Research. In addition, he is a professor in the Department of Biomedical Science at the University of Missouri, Kansas City School of Medicine. Today, Dr. Koulen will discuss the topic of the synergistic mechanisms underlying the early-stage alterations in renal function following experimentally induced dyslipidemia and hyperglycemia. Please join me in extending a warm welcome to Dr. Peter Koulen. Okay.
Thank you so much, Dr. Kim. Let me share my slides. Can you see them okay?
Yeah. We do.
All right, very nice. And I will, for the presentation, I will just briefly stop the video if we have any connectivity issues. And then I heard that you typically ask questions at the end, but if there's something really urgent that needs clarification, feel free to interrupt me. So thank you again for the nice introduction and for inviting me. I know that there is significant interest in diabetes research. We are at the University of Missouri, Kansas City School of Medicine, very interested in that in a much broader sense than just today's presentation. We have a strong emphasis on rural medicine, as well as also a focus on diseases that affect our urban communities. And for that same reason, we have a strong area of emphasis in that field. My research group is just covering a smaller aspect of that. And I wanted to give you a brief overview of what I'm going to talk about today. One of the reasons why we're interested in a study linking dyslipidemia with diabetes complications such as diabetic retinopathy is a simple population statistics or epidemiology phenomenon that we see in Kansas City. We just have an exploding caseload of individuals that present with both of them. A colleague of mine in the School of Pharmacy, Dr. Tom Johnston, and we thought, let's look into conducting clinical studies in that field. And we ran into several roadblocks. The most important one is that we can't really distinguish dyslipidemia and diabetes very nicely in patient populations because we don't have any patient populations in Kansas City that fall into one or the other category. They typically fall into both simultaneously. So this led us to developing an animal model, and that animal model is going to be the topic of today's presentation. What we found is that both disease-causing mechanisms or both diseases affect diabetic retinopathy development and outcomes synergistically, not just running in parallel like one would expect. So when we talk about diabetic retinopathy, you see here the picture of those kids playing soccer ball. That's what a normally sighted individual would see. A person with diabetic retinopathy in the end stage would see something like this. So the areas of the visual field have dropped out. You have severe damage to the retina and What you can also see is that everything is a little dimmer and fuzzier. So in addition to these dramatic dropouts of visual field components, you also have an overall diminished visual performance in those patients. So when we talk about diabetic retinopathy, typically on the clinical side, what is being discussed is that this is a vascular disease, so that blood vessels in the eye are affected and they're causing a lot of the problems. Statistically, we see in the nation something similar that we also observe in our microcosmos in Kansas City. This is an older study from 2010, where they're projecting that by 2050, we're going to have 14.5 million diabetic retinopathy sufferers in working age adults. And unfortunately, there are a couple of issues with diabetic retinopathy that make this situation even more complicated. There's no distinction between type 1 and type 2. Type 1 individuals actually are oftentimes at higher risk because they usually have a longer time that they spend with diabetes. Unfortunately, for both, there are no early symptoms. There are ocular diseases where you notice changes in your vision. You see in a physician, and they typically can help you. In diabetic retinopathy, similarly to glaucoma, there are no such early symptoms, and that's why early detection is necessary. And my study that I'm going to present today is exclusively focused on this early aspect of the disease, where if we can intervene at this early time point, we have actually a good chance of catching things and treating things that are helping us to prevent this. So this older study that I mentioned, where we see this increase or doubling almost of diabetic retinopathy individuals from 2020 to 2050, this is basically a fraction of the 80 million people that are estimated to be in the age range of 65 and older in 2050. And as I said, it's the #1 cause of vision loss in working age adults. That is slightly different when we look at the whole population or if we look at the older population, but it is from a healthcare and economic impact, a disease that cannot be ignored. When we look at the phenotypes of the disease, what physicians typically see early in the disease is no significant changes, but a swelling of the area of highest visual acuity, which we call the macula. If we do more sophisticated analyses that are typically not part of clinical diagnostics, at this early stage, we can already see a loss of pericytes in the blood vessels, a thickening of the basement membrane, and similar things. What is especially interesting for me, that at this early stage and basically progressing throughout the whole disease development, we see a loss of the cells that communicate between retina and brain, the so-called retinal ganglion cells. In the higher stages of the disease progression, we then start to see phenotypes that are typically associated with vascular pathology, like aneurysms. We see bleeding, and we see proliferative diabetic retinopathy, which means that there's new blood vessels forming in areas where blood vessels have been lost to basically make up in the lost oxygen supply. However, these newly formed blood vessels are ineffective in two ways. First of all, they don't do the job that blood vessels do, which is to provide sufficient oxygenation of the tissue. And on top of that, they're typically leaky, and they lead to additional swelling of the retina and hemorrhages. And when you think back about the picture of the two soccer-playing kids and what a diabetic retinopathy patient sees, whenever you have bleeding in the retina that's visible in such a fundoscope image, that would be a black spot that the patient can't see the environment anymore. So when we look at diabetic retinopathy, the focus is typically on diabetes, on hyperglycemia, and what is oftentimes ignored, other aspects of the metabolic syndrome, which typically precede diabetes. So we looked into the clinical literature and looked at this from various angles. So when you look at clinical studies that indicate that dyslipidemia, so abnormal lipid levels or lipid compositions, can already lead to retina and visual function impairment. So, for example, during metabolic syndrome or insulin resistance without properly diagnosed diabetes, we already see an increased risk for retinopathy in an epidemiology perspective. Hyperglycemia in infants has also been linked with an increased risk of retinopathy of prematurity. That is a very small number of patients affected. However, it's a very interesting indicator that there's potentially shared pathways. When we look at evidence for diabetes impairing retinal function, the literature is vast. So typically what we can see is that increased levels of oxidative stress are seen. We can see that individuals with higher levels of aldose reductase have an increased risk for diabetic retinopathy development. There were even prospective studies where hyperglycemia has been directly linked to changes in the vasculature, but also to a large number of other mechanistic pathways, such as advanced glycation end products, glutamate toxicity when it comes to neurotransmission, and also various aspects of lipid peroxidation, which lead to neuronal damage. There's a smaller number of studies that looked at synergistic or additive evidence for diabetes and dyslipidemia causing visual impairment or retina dysfunction. So in diabetic patients with dyslipidemia, there's a significantly increased risk for worse outcomes of retinopathy. In the US, elevated HDL, for example, was found to be protective against early changes and has been investigated as a strategy to combat that. Whereas when we look at diabetes patients with diagnosed diabetic retinopathy, their greater levels of ApoA1 in the vitreous have been found as a key distinguishing factor when we compare those to non-diabetic controls. There were several prospective studies. There are also interventional studies that have had varied outcomes. In diabetic patients that do not show signs of diabetic retinopathy, dyslipidemia is typically a good predictive factor. The same is true for ApoA1 if we focus on vascular dysfunction. And patients with dyslipidemia are typically at much higher risk for these early complications. When we look at shared pathophysiologies between dyslipidemia and diabetes, when we look at diabetic retinopathy, there are again many. The most interesting ones are lipid peroxidation, serum malonaldehyde, and also a concurrent dyslipidemia in poorly controlled diabetic patients. Many of the inflammatory factors or pro-inflammatory cytokines have been identified, and as indicators of both diabetes and dyslipidemia worsening. As a intervention strategy, omega-3 fatty acids have been identified as decreasing oxidative stress, and that's now an FDA-approved strategy. And there is a suggested overlap of pathologies based on the interventional success of that strategy. Several studies have suggested that controlling dyslipidemia, for example, with statins, may protect against diabetic retinopathy. But as I've mentioned, there's quite a large number of these studies, and some show benefits, some show a lack of benefit. You can take a lot of that information and reduce it to pathways that are interesting for basic science strategies that are either in the drug development field or in the drug target discovery field. And while there are several pathways that are very typical for dyslipidemia, that are oftentimes associated either with a direct damage of nerve cells, such as increased INOS activity or a beta accumulation, leading to a damage of the blood-retina barrier. There are also other factors that are more typical for inflammation affecting glia cells in the retina. On the diabetes side, there's also many factors that you're probably well aware of that not only cause damage in the retina itself, but also elsewhere in the system, and that cause a lot of the pathophysiologies that we are aware of with diabetes. But what was astounding is the large number of pathways that are shared between the two pathophysiologies, and that are very wide-ranging when you think about very fundamental signaling pathways like PPAR alpha receptors, FGF signaling, but also the varied pathways that affect the microvasculature and that share a lot of common pathways, for example, with cardiomyopathy subsequent to diabetes. In addition to that, many of the clinical studies also point us in the same direction that we were interested to conduct our basic science study, which indicate that some aspects of lipid signaling are protective and some aspects of lipid signaling are aggravating or enhancing diabetic complications. And our goal was to design a study where we could quantify retina function. And in order to do that, we scoured the literature for what would be a clean diabetes model. And as many of you are probably aware, that is very difficult to find in terms of a model that would allow us to really recapitulate key aspects of type 2 diabetes in humans. And we would, once we've identified such a model, which is the DB mouse, there's some modest disturbance of the lipid profile in these animals, but they're as close to a wild type as you can get if you still want hyperglycemia. And we combined that with a model that my collaborator, Dr. Johnston, had developed now almost 40 years ago, where dyslipidemia was induced chemically. And our goal was to then be able to combine the two, as well as to look at effects individually and to test if there was additive or synergistic effects on retina and visual function in diabetes. So we used 2 cohorts of male mice, and preempting a question that will likely come, we're currently in the process of repeating the same study with female mice, but we're just a small basic science lab. We don't have the facilities to run large cohort studies like industry, but that will be another study that will come out soon. And these mice were divided into 2 groups. So the wild type C57s and the DBs on a C57 background. The DBs are a leptin receptor mutation that leads to obesity-mediated diabetes. And one group in each of those strains received poloxamer 407. Here's the structure of that. It's a very large polymer, and what always scares me about this molecule is we're using that to induce dyslipidemia, and how frequently this compound is used in everyday applications in the chemical and pharmaceutical industry. So we administered this compound intraperitoneally in both the diabetic and the wild type animals, and then we had saline controlled in both to give us a clean strain control and a clean diabetes control. So what we did was to measure blood glucose, total cholesterols, triglycerides. We measured retina function, which is basically just focused on the retina itself. So that's not measuring vision, it's just measuring how well does the retina component of our visual system work. And then we also measured visual function overall using a reflex measurement that I will talk about in a little while. But it's basically a behavioral assessment that allows us to go back to the animals multiple times without having to do any invasive measurements. And it uses a reflex that's arises from an interaction of the retina with the brain. So we're measuring both how well the retina processes information and how well the brain turns that into a motor response. So to give you a basic characterization of these animals, we started with 2 months old animals, and we observed them over a period of 1 month, a relatively straightforward paradigm. But you have to keep that in mind. We're not talking about aged animals or animals that are significantly impaired. We're really talking about early stage animals that have, however, significant hyperglycemia in the DBs and in the diabetic animals. And that is also maintained in the animals that have induced dyslipidemia and have the same genotype. So they have a pretty high blood glucose level of around 500 mg per deciliter. When we look at the lipid profile of these animals in a very high level look at triglycerides in general, what we find is that very consistently by interfering with metabolic pathways that lead to elevated triglyceride production using our chemical induction approach, we get a very significant elevation of triglycerides in our induced animals, in both the wild type and the diabetic animals. As I mentioned earlier, the diabetic animals have a small lipid disturbance, but it's significantly less than what we see in, for example, animals that are being fed a high-fat diet. And statistically, the triglyceride levels are not different from wild types. However, in our diabetic animals, we see that have been chemically induced for dyslipidemia, we see a very large elevation that is even higher than just in the wild types. And there's already an interesting aspect that the signaling pathways that are triggered in wild type animals by the chemical induction are already primed in diabetic animals to be even higher elevated. When we look at cholesterol values, that's even more pronounced. So in the presence of our induction chemical for dyslipidemia, we see a strong increase of total cholesterol that is also present in our wild types, but much stronger in our diabetic animals. And when we look at the distribution of different lipids that contribute to total cholesterol, we see that in our induced animals, the HDL component that has been identified in humans as a protective aspect for diabetic retinopathy is reduced in the chemically induced animals. So both the wild types as well as the DBDB animals. The lipid profile with regards to cholesterol in the diabetic animals, animal controls, is relatively normal. We, however, see a moderate increase in VLDL. That increase is much more pronounced in the dyslipidemic and diabetic animals. So when we measure visual acuity and contrast sensitivity, or in more general terms, vision, we are faced with a similar problem that pediatricians or pediatric ophthalmologists are faced with. You can't just tell the animals, read the chart on the wall. So we have to come up with more sophisticated ways to measure that. And what we're using is called the optomotor reflex. In humans, the optomotor reflex can also take on the form of the optokinetic reflex. So when presented with a moving stimulus, either the whole body, the head, or just the eyes follow that stimulus. And that reflex, you might have observed that on yourself when you're stopped at a railroad crossing and a train goes by and your eyes follow the train or your head follows the train. So the optomotor reflex means that when we rotate this striped pattern that's presented by these 4 computer screens, the mouse sits here in the middle, and here we look at this from the top. When these striped patterns move in a certain direction, the mouse then reflexively follows that. And as soon as the mouse follows that, we call that tracking. We know that they detect it. We can measure both the left eye and the right eye separately due to the rodent anatomy. In humans, we wouldn't be able to do that, but in humans, we can tell them to cover up one eye. What we can extract from that is both the contrast sensitivity, so that would mean how dim can we make these black stripes and they can still see them, and how accurate is their visual acuity, so how thin can we make those stripes, or how close together can we make those stripes and they still detect them. So me explaining that is fairly complicated. Here you can just see it. So this mouse is presented with this stimulus, and there's a little crosshair on the fixed on the head. And you see when the mouse is tracking the pattern, it's moving along. And we can then mark that as detected and switch to the next frequency or intensity. This was always a lot of work for postdocs and students in the lab. We've now have a system that's fully automated, including a reminder system for the mouse when it falls asleep during the test. In the past, you would just knock on the box, and now that's also computer controlled. So data acquisition has gotten a little easier. So what we find in these animals, and this is kind of a busy slide, but the most important aspect is pretty much shown here in the first part. The visual acuity in the animals that are diabetic and dyslipidemic is significantly reduced, and the same is true for the contrast sensitivity. However, in all of these animals, visual acuity and contrast sensitivity are correlated, just like they are in the wild type. The only exception, and that's the same also in humans, they're negatively correlated in diabetes-only patients. That's likely an indicator that we're dealing with significant changes at the level of the inner retina. So when we look at additional correlations, so here in the top panels, we look at correlations between blood glucose and visual acuity and contrast sensitivity. And here in the middle, we look at cholesterol, and down here at triglycerides. The interesting aspect for all of these is when we look at these downward facing triangles, they're always set apart from the rest of the animals significantly in that their worse physiological parameters are typically associated also with worse performance. Visual acuity, as being the more physiologically sensitive measure, typically gives us a faster short-term answer. If there are changes in contrast sensitivity, those are more profound and higher impact. And as I mentioned earlier, our model is an early stage disease model, so we would not expect too many changes there. The second technology for non-invasively measuring activity of the visual system is electroretinography. And I'm showing you the human applications first. When I was a graduate student, it did not look as easy as this. It was basically the whole room was an electroretinography studio. It took quite a long time to attach electrodes. It was very tiring for the patients because a lot of stimuli were presented. We now have a handheld device that gives us answers to very simple ERG stimulus patterns in a matter of minutes. It's very convenient for the patient because there's no drugs they need to take or injections or anything like that. The electrode is just a little band-aid under the eye. There are more sophisticated ERG systems that are on the market, especially when there are more detailed questions being asked with regards to different cell types, different types of vision, such as color vision versus night vision, etc. But for our purposes, oftentimes a simple flash to the eye, light flash to the eye that elicits an electrical response of the neurons that we can detect with these electrodes is sufficient. In a mouse, principally that's the same. The only problem is they usually don't agree with having a camera held to their eye. So they're under anesthesia. They have this little anesthesia nose cone here. And then we have electrodes directly placed on the cornea with little contact lenses to give us high resolution electrical contacts. That used to be the case in human patients too. And then we have reference electrodes. It's a relatively involved setup, but it takes only about 30 to 40 minutes for acquisition of a full spectrum measurement. And the animals return to their home cage and have no negative effects from this non-invasive measurement. This system also has been used in larger animals. We've used it in rabbits, and there are some of my colleagues who use it routinely in dogs. Veterinary ophthalmologists want to know if they do cataract surgery, is there actually a retina functioning behind the cataract that they're removing, or are we wasting our time? And there's similar systems used in horses, orangutans. There's a long list of applications. So when we measure electroretinograms, we typically get a defined response. If you look at the black line here, that is typical for both the stimulus and the response of the retina. When we look at our experimentally altered animals, which are here in dotted and red lines, you see that some aspects of the pattern stays the same, but that both the temporal component here on the x-axis, so that would be time in milliseconds, and also the amplitude that's measured here in microvolts can be changed due to disease patterns. And what we see in our animals is that under the conditions of both dyslipidemia and hyperglycemia in our DBDB P47 treated animals, we see a strong reduction in the A wave time, the duration, as well as in the amplitude. And there's only a reduced effect on the B wave time, however, still a pronounced effect on the B wave amplitude. So when we graph those as populations, and correlate that to visual acuity measurements, what we find is that both at the system level, so when we talk about retina and brain communicating properly with each other, as well as at the retina level, there's a reduction or an impairment predominantly for the dyslipidemic hyperglycemic animals. And you can see similar things for contrast sensitivity, even though not as pronounced. However, very pronounced when you correlate that with the B wave amplitude. You get a nice clustering of the animals that are affected by both disease-causing mechanisms. And when we look at the synergism of these features with regards to the scotopic A wave, which would be vision under dark conditions or low light conditions, we again see that while there is typically a reduction in the A wave time, the correlation with low visual acuity is fairly pronounced, and the same is true for B wave time and visual acuity. With contrast sensitivity, as I pointed out earlier, that's a late stage disease marker. Those correlations are less pronounced. So when we look at B to A wave ratios, which are typically the values that, for example, pediatric ophthalmologists use to assess the function of a retina in individuals with genetic disorders, we see that in the dyslipidemic and hyperglycemic animals, we can also see a change in the B to A wave ratio under light conditions, where it's already fully expressed under dark vision. So when we break this down with other stimulation paradigms and look at the photoreceptors that are contributing to low light vision, we see a decrease in diabetic animals and a further decrease in dyslipidemic diabetic animals. The same is true under other stimulation paradigms, where we look at a mix of low light level and ambient light levels. And if we go to the aspects of color vision, those differences are less pronounced. This is mimicking very nicely what also goes on in human patients, where typically first changes are observed in low light vision or scotopic vision, and subsequent changes occur later in the disease development with photopic or color vision. When we look at how these values are correlated with disease parameters that affect both glucose and lipid levels, we can again see our downward triangles sticking out as clusters that are by themselves, that show a both the feature of an elevated disease indicating phenotype, as well as a lower performance in the function of the retina. So this animal model that we've been working on shows a clear elevation of lipid levels that are indicative of dyslipidemia. What we found also is that the resulting hyperlipidemia is much more pronounced in diabetic mice, and that the reduced visual acuity that we saw for diabetic and dyslipidemic mice was more pronounced than for either condition by itself. The ERG measurements told us that especially under low light conditions, the retina function showed a significant decline in key parameters that point towards the inner retina, and that this was already the case in dyslipidemic animals, but more pronounced in animals that had dyslipidemia and diabetes. And these strong correlations between retina impairment and total cholesterol and triglyceride levels were indicative that the disease process started at the front end of the visual signaling pathway, which is the retina. So when we look at the severity of retinal dysfunction in diabetes, what we see is that the presence of hyperglycemia and dyslipidemia can act synergistically and impact retinal function. And this model of a combined hyperglycemia and dyslipidemia can allow us to dissect these features further. As you've seen, we can, for example, analyze individual cell components in the retina, but it also allows us to be more targeted in our therapeutics development approach, where we look, for example, at combination strategies to lower bad cholesterol and control hyperglycemia simultaneously. I wanted to thank all my collaborators, but specifically Dr. Tom Johnston, who's a pharmaceutical scientist in the School of Pharmacy, and who's also a member of our Vision Research Center. And in my lab, specifically Dr. Gina Edwards, who's been key for developing the mouse model and developing the non-invasive assessment of vision and retina function. And I also wanted to thank our funders, and would be happy to answer questions at this point.
Thank you, Dr. Koulen. This was an excellent presentation. Can you hear me?
I can hear you fine, yeah.
Yeah. Can I ask you a question about the objectivity of collaboration, for instance? You are there, you were in KC. Is there any cross-collaboration on this specific research between your team and the folks here in ophthalmology in the School of Medicine in Columbia?
Oh, in the ophthalmology department there? Not on this particular project, but for example, we set up a joint annual symposium where we meet. We have the pleasure and privilege to exchange our trainees every now and then, and to have a very good interaction at the fellows and residence level. For this particular study, as I said, this is a pilot study that we started here in Kansas City that was born out of a conundrum that we faced in the clinics. But we're hoping to find drug development specialists and people that are interested in peripheral and systemic effects of diabetes. We are now in a much better position clinically to try and advance this again into a clinical study, and then ultimately, hopefully, also into clinical trials. But the advantage of the mouse model is that we can ask very specific questions and also titrate disease parameters, in that we can control the extent of both disease development processes. So the short answer is, we haven't collaborated on this project, but we had interacted on other projects.
Yeah, this project, it clearly shows that you basically setting yourself up for a trial on humans, if you could, because now all the literature that you just presented pointed to this relationship strongly. And not surprisingly, 99% of the diabetic patients died of heart attack at a much earlier age than the life expectancy.
Yeah.
But the retinopathy is always suspected for many years, but now it's really proven. I mean, the data you gave proved that that's synergistic effect. Mm-hmm. And that synergistic effect also points that clinical medication for dyslipidemia may affect tremendously the proclivity of developing diabetic retinopathy. So a trial on that's going to be very interesting for any pharmaceutical industry to do. I don't think you're going to have a trouble getting. That's not really
I strongly agree, and there's recent studies out that you can actually use the retina microvasculature as a surrogate marker of cardiovascular health, and you can use morphological parameters of the retinal vasculature as a predictor of cardiovascular disease outcome. So that was very interesting for us, and the advantage is it literally takes only a few minutes to acquire a decent fundoscopic image. However, many of the cardiovascular diagnostics are much more involved and much more detailed. So, and when we look at some of the signaling pathways, there's perhaps not surprising, but surprising if you've never thought about it, shared signaling pathways in terms of inflammatory signaling, in terms of, I don't want to call it neuronal degeneration, but I would call it the degeneration of excitable cells. And something that's very often overlooked is the effect on vasculature, parasites, and changes in the vasculature morphology.
That's amazing. One curiosity I have, it's about how do you figure out how to automate the reminder to the little mice to not let him sleep? Is that done, I would say, automatically? So it's a program, is that a software that you created, or?
Yeah, it's not created by us. It's the inventor is Dr. Prusky. He is, let me think, I always think he's in Canada, but he recently relocated to, I think, SUNY. Yeah, he is an expert in behavioral assessment of vision, and we've been collaborating with him for many years. We were among the first to look at the impact of disease phenotypes on these reflexive behaviors. So we published, for example, on glaucoma in that field. The reminder is very simple. The software just plays a little buzzing sound and that wakes them up. But you also have a person that runs the experiments, and they can manually also interfere. It happens to the best of us that during an activity you sometimes fall asleep, and the mice are no exception.
Listen, I'm gonna have to leave because I have another meeting exactly in 1 minute from now. And Dr. Kim just remind me of that. That's also the diabetes grant that we have. That you're part of. This presentation is part of it, is to share all the knowledge we have on diabetes, not only from us, but from others who can bring, we can bring to the network of research at